Hi everyone, I hope you're enjoying your holiday break. This lecture is going to cover material from chapter 15, Life Near the Surface. So we've spent the last few lectures talking about benthic organisms and benthic habitats. And today we're going to switch over to talking about organisms and food webs found in the water, starting with this epipelagic region, which is also sometimes called the euphotic zone or the photic zone. So this is the upper layer of the water column where we have sunlight. So from the surface of the ocean down to about 200 meters is the epipelagic region or the euphotic zone, the photic zone. And that's going to be the focus of this lecture. So within the euphotic zone or the epipelagic zone, we have phytoplankton, zooplankton, and nekton. Remember, plankton are drifters. So they're able to move about a little bit, but they're not strong swimmers. They're not going to be able to swim past a moving ocean current. So these are generally smaller organisms. And the nekton are strong swimmers. So these are bigger organisms and marine fishes, marine mammals that are able to swim through the water. Two different types of plankton we've learned about, the phytoplankton. So phytoplankton are able to photosynthesize. These are the autotrophic drifters. And zooplankton, which are the heterotrophic drifters. And the epipelagic region is really important because it's providing food for deeper habitats. So dead organic material from the epipelagic zone sinks down to the seafloor and feeds deeper layers. So we've talked a lot about detritus as a food source for benthic organisms. And some of that detritus can be coming from production in the epipelagic zone. And the epipelagic zone also provides food for many different species of fishes, marine mammals. Seabirds will feed from the epipelagic zone, and humans harvest fish, or industrial fishing operations are focused on this epipelagic zone. Different size fractions of plankton that we see in the ocean. The very, very smallest are called femtoplankton. Then we have picoplankton, nanoplankton, and microplankton. So these smaller size classes from microplankton down to femtoplankton are so small that we need a microscope to see them. The microplankton, mesoplankton, and macroplankton are grouped together in a group called net plankton. So these organisms are large enough that we can filter them or collect them from the water using a net. So here's a nice underwater shot of a plankton net being towed from a boat. The microplankton, mesoplankton, and macroplankton are large enough that they're going to be captured in the net. They're not going to go through these tiny pores. So a larger microplankton, you might be able to see. Mesoplankton and macroplankton, you're able to see without a microscope. And then we have the megaplankton. So these are jellyfishes and seaweed species that are larger, but they're still not able to swim, so they're considered plankton. So we've talked about surface area to volume ratio, and having a large surface area to volume ratio is beneficial because surface area is where we have exchange of nutrients and waste. So having a large surface area to volume ratio means you have a lot of surface area to exchange nutrients, but a smaller volume to take care of. So the smallest plankton species are going to be better able to survive in really low nutrient waters. So we've seen this image before of chlorophyll concentrations in the global ocean. They're really low chlorophyll concentrations. These low nutrient regions are places where we would expect to see picoplankton, really small plankton. And these are single cellular organisms, so small that we need a microscope to see them. The net plankton, the larger size fraction that we can capture with a net, most of which we can see without a microscope, Net plankton are going to be found in regions that have more nutrients. So where we see a lot of chlorophyll, we have high nutrient concentrations, and that's where we're going to see this larger size fraction of plankton. That can be in upwelling regions or close to the coast, anywhere where we have more nutrients. But the smallest plankton, the pico, the nano, the femtoplankton, are going to be better able to survive out where nutrient concentrations are low because they have that high surface area to volume ratio. The nanoplankton, picoplankton, and femtoplankton are single cellular plankton species, and they're mostly bacteria and archaea. Though with the nanoplankton, we get some eukarya as well. So the bacteria and the archaea domains include single cellular organisms that are very, very small. They make up most of the picoplankton. Viruses make up the femtoplankton. And then we have some single cellular eukaryotes that are included in the nanoplankton. The net plankton include eukarya. So these are some phytoplankton, some zooplankton, as well as meroplankton. So remember, the meroplankton are organisms that are plankton in their juvenile stage. When they develop and grow into adults, they become nekton. But in their larval stage, they're not able to swim, so they're considered plankton. And those might be captured in a plankton net. So these larger size fractions, the macro, meso, and microplankton that are included in the net plankton, some are phytoplankton, some are zooplankton, some are meroplankton, and they're coming from the eukarya domain. So we might have some organisms from the kingdom Protista. So remember, the heterotrophic protist we call protozoans, like radiolarians. And the autotrophic protist we call algae. So we can have both single cellular algae and multicellular algae. But there are many other kingdoms in addition to Protista that we might see in the net plankton. So Cnidaria, Arthropoda are a couple examples, especially in the megaplankton, these larger species as well. But the larger plankton species are coming from eukarya. The very small single cellular plankton species are coming mostly from bacteria and archaea, as well as viruses. So we'll go through these different size fractions and use some examples that we've seen before. So these are examples of species that we covered in chapters 5 and 6. So the smallest femtoplankton that are less than 0.2 microns are mostly viruses. So remember, these are non-cellular organisms that can insert genetic material into a bacteria. The nanoplankton and the picoplankton are mostly bacteria and archaea. So from the bacteria and archaea domains, they can include methanogens and trichodesmium, the cyanobacteria colonies. The net plankton, the larger plankton species, include algae. So we talked about diatoms, dinoflagellates, coccoliths, and different seaweeds. So these are all members of the Protista kingdom, the eukarya domain, and some protozoan species as well, the radiolarian and the foraminifera. Few species that we haven't talked about yet are copepods and krills. So these are from the Arthropoda phylum, and copepods make up about 70% of net plankton species. Krill look similar to copepods, but they're a bit larger. So the copepods are less than 20 millimeters. Most of these you can see without a microscope. The krill are larger, so anywhere from 2 to 20 centimeters. And these are the species that are consumed by whales. So baleen whales will feed on krill. The copepods are a little too small for these larger marine mammals to see and to hunt. But fishes, whales, and seabirds are able to see these larger krill species and eat those krill. Both the copepods and krill are primary consumers that feed on plankton. Some other net plankton are salps and larvaceans. These organisms look like jellies, but they're actually not part of the Cnidaria phylum. These are part of the Chordata phylum and the subphylum Tunicates. So these are tiny organisms that secrete a mucus net. They're able to get rid of this mucus net. So here's the net here. On the inside is the organism. So on the right, we're looking at a salp. This is an individual salp, but they're often found grouped together in these long chains. So this organism is able to secrete this clear mucus net and then pump water through that mucus net and filter plankton from the water. And the larvation operates in a similar way. So these are filter feeders and these are net planktons. So pretty amazing organisms. And another example here of how the epipelagic region can provide food for deeper layers of the water column and to provide food for the benthic, the sea floor. And this footage is coming from MBARI, the Monterey Bay Area Research Institute, right down the road. Some other plankton species include jellyfishes, the ctenophore comb jelly, arrow worms, and my favorite fish, the ocean sunfish. So this fish can move around, but it's not a strong swimmer, so it's often grouped into the plankton. And then we have nekton, the strong swimmers, and there are many different species of nekton found in the epipelagic. We have invertebrates, vertebrates, reptiles, marine mammals. The nekton tend to be mostly third and fourth level consumers, so they're eating other nekton. But there are some smaller nekton that are primary or secondary consumers, or nekton that eat plankton. So whales are an example. Whales that are feeding on krill would be a secondary consumer. But a lot of the nekton we see in the epipelagic are these third and fourth level consumers. Organisms in the epipelagic need to stay afloat. So we have a lot of phytoplankton and primary producers, and they want to stay as close to the surface as possible to get the most sunlight. And because these phytoplankton are forming the base of the food web, organisms, the secondary consumers that are feeding on the phytoplankton, also want to stay towards the surface because that's where all the food is. So there are a number of different strategies that organisms use to stay afloat. They can increase their surface area, or they can increase their buoyancy. So when we talked about phytoplankton, we saw all of these different shapes and sizes. So different spines and projections that these phytoplankton have can help them stay close to the surface. So by increasing their surface area, by having these long spines and projections, they're better able to float. Sometimes we see these species clustered together. So this is another way to reduce drag. So increasing surface area will reduce the drag and help these organisms stay afloat. And then some organisms like jellyfishes have a parachute shape that help them float. Some organisms secrete lipids or oils to increase their buoyancy. So we talked about this when we learned about diatoms. So the diatom is able to produce these lipids inside its body, and that helps increase buoyancy. And then fishes have swim bladders. So they're able to fill this swim bladder with gases to help float and stay near the surface. There are some organisms called pleuston that actually rest above the surface, like the Portuguese man of war. So they have gas-filled structures that actually let them rest above the ocean surface, and then they can use wind to move around. In the epipelagic, we see a couple different strategies for camouflage or blending in. Some organisms are transparent. So the copepod and the salp have this transparency that helps them blend in with the surrounding water, especially near the surface where it's nice and bright. They're better able to blend in. Other organisms use what's called counter shading. So the ventral or the bottom surface is a light color, a white or a silver, and the dorsal surface or the back surface is a darker color. So if we're looking up at a penguin, we're on the seafloor taking this picture of a penguin, has this white stomach that's going to help it blend in with the light at the top, the light at the surface of the ocean. But if we were standing on a boat looking down on this penguin, we would see a dark back that would blend in with the sea floor. So looking up at this tuna, this white underbelly would blend in with the bright sunlit surface layer, and looking down on this tuna, the dark back would blend in with the ocean floor. So this is an adaptation that helps these organisms avoid predators. Most nekton in the epipelagic are strong swimmers. So we talked about tuna being able to migrate thousands of miles across the Pacific Ocean. And they have a number of adaptations to be able to do this, to be able to swim fast and to be able to swim these long distances. So first adaptation is having this streamlined body shape to reduce drag. We talked about myoglobin. So these fishes are able to store more oxygen in their muscles, and that helps them swim for long periods of time. So they have red muscles for sustained swimming, and they also have white muscle for short bursts of speed. They also have this really efficient transfer of heat between blood vessels. So they want to keep those muscles warm so they're able to swim these long distances. So blood that's flowing, so here's the core of the organism. This is kind of a cross section of thick muscle tissue. Moving away from the core of this organism out towards the skin, we're going to be approaching colder seawater. So the blood that's moving towards the skin is going to cool down and then be pumped back towards the core. So what happens is blood that's on its way to the surface is going to get rid of that heat. It's going to transfer that heat to a blood vessel that's carrying blood back to the core. So this blood that's flowing out towards the skin is going to lose that heat anyway. So rather than losing the heat through the skin to surrounding seawater, it's going to transfer that heat to a blood vessel that's returning blood towards the core. So rather than lose that heat to the exterior of the body, they're going to transfer it and keep it within the core of the body. So this really efficient heat transfer within the blood vessels helps keep those muscles warm, and that enables these fishes to swim long distances for long periods of time. So some of these fishes have been tracked swimming 40 miles in a day. So let's look into this streamlined body shape a little deeper. So streamlined organisms have less drag, and that makes them faster and more efficient swimmers. So as a fish is moving forward, we have water particles that are flowing over the body. And behind the body, we get some turbulence. So you can think of this as the wake that you might see behind a boat. So that boat is moving forward, the water particles are flowing around that boat, and then we get this turbulent wake behind the boat. The more round that boat is, the more of a wake we're going to see, or the greater the turbulence. But if we have a streamlined shape, we're going to see less turbulence or less of a wake. So organisms with this streamlined shape, water particles are going to move around the body, and then there's going to be less turbulence behind the body to slow them down. So having this streamlined shape makes them faster and more efficient swimmers. These fishes have other adaptations to help them be fast swimmers, and you can think of an Olympic swimmer. So Olympic swimmers wear tight suits. They shave hair off their legs and their arms. They cover the hair on their head with a swimming cap, and they have this streamlined form that helps them swim faster through the water. So fishes in the epipelagic have a lot of adaptations that help them swim fast through the water as well. So we talked about the streamlined bodies. Many of these fishes have very few scales. So those scales are something that can create drag and slow them down. Without the scales, they're able to swim faster. Their eyes are very close, are flush with the body, which means they sit very close to the body. And they even have a transparent eyelid that's thought to help reduce drag. Their fins are very stiff and narrow. And some fishes, like swordfish, marlin, and the sailfish, which is pictured here, even have these long bills that help them cut through the water. Zooplankton in the epipelagic zone are able to migrate vertically. So remember, plankton aren't strong swimmers, but they're still able to move around a little bit. So they're still considered plankton because they're not able to swim past a current. They're not strong swimmers, but they're able to migrate up and down in the water column. And they do this during the day and during the night to avoid predators. So during the day, when the surface water is nice and bright and sunny, they actually migrate deeper below the euphotic zone, so usually below 100 meters. And they do this to avoid predators at the surface. So seabirds flying over on this bright sunny day can easily see the zooplankton and feed on those zooplankton. So they go down deeper in the water to hang out and hide. And then at night, they come back to the surface to feed on these primary producers. So they're more difficult to see in the dark. These phytoplankton have been reproducing and growing all day long in the sunlight, and they're going to come up and feed at night. So in this example, we have copepods and krills, but other zooplankton species do this as well. They migrate deeper during the day to hide from predators, and they come back to the surface to feed at night. And this was discovered using an echogram. So we've talked about echo sounding and estimating the depth of the seafloor, where we send a sound pulse down into the ocean and we record the amount of time it takes to reflect back to the ship. So in a similar matter, scientists have discovered these different layers of zooplankton moving more shallow and deeper throughout the day. So this is data looking at sound pulses, sound scattering, and on the bottom we can see the time. So during the nighttime, we see this layer of zooplankton near the surface. So they're coming up at night to feed when they're harder to see. And then as the sun comes up, they migrate back down deeper. So they're going to hide out during the day to avoid their predators at the surface. Epipelagic food webs can be quite complicated. So some species of nectin feed on different prey throughout their lifetime. An adult herring is eating sand eels, amphipods, larvaceans, but during its early life stages, it might be feeding on jellyfishes. So the type of food that adults versus juveniles eat changes. And many epipelagic organisms eat a variety of prey. So this adult herring can have many different trophic levels depending on the pathway that we follow. So an adult herring that's consuming larvaceans, we have primary producers. This is a primary consumer, and that would make the herring a secondary consumer. But if we follow a different pathway, so we have primary producer, primary consumer, secondary consumer, third level consumer. Now the adult herring is a fourth level consumer. So depending on the prey, this adult herring can have many different trophic levels, and that adds complexity to this epipelagic food web. And in the epipelagic, the microbial loop is really important. So phytoplankton, primary producers, remember they're making organic matter through photosynthesis. So they're using energy from the sun to combine carbon dioxide and water and produce organic matter and oxygen. But about half of that organic matter is actually lost to the water. And the microbial loop is really important in saving or recycling that organic matter. So when that organic matter is released to the water, we call it dissolved organic matter, or DOM. So it's dissolved in seawater. We can't see it, So we know that there's sodium and chloride and different salts dissolved in seawater. If you're looking at seawater or touching seawater, you don't actually see or feel those salts, but we know that they're there. We can measure the salinity, can evaporate the water and actually precipitate the salt, but the salts are dissolved. We can't see them even though they're there. So this organic matter is released from primary producers into the water. And even though we can't see it, we can measure it using different chemistry tools. So we call it dissolved organic matter because it's dissolved in the water. We can't see it. So a lot of organic material produced by primary producers is released to the water. It's dissolved in seawater. And the microbial loop recycles that or uses that organic matter. Organic matter is released to the water sometimes by leaking out of cells. Other times it's released during grazing. So in this image, copepods are feeding on diatoms, but they're sloppy eaters. So imagine sitting down and eating a really crunchy croissant. You might spill pieces or crumbs of that croissant on the table. So in a similar fashion, the copepods are feeding on zooplankton. Copepods are feeding on diatoms, and they're not eating the diatom in one bite. So they're taking multiple bites, and they're spilling crumbs of organic matter that gets released to the water. We talked about viruses, so the bacteriophages. Those are viruses that can insert their genetic material into a bacterial cell, bursting that bacterial cell and releasing its organic contents, the organic matter. And organic matter is also excreted as waste. So fecal pellets from these copepods are going to release this organic matter back into the water. So dissolved organic matter is released by grazing, that's sloppy eating, spilling crumbs, can leak from cells, be released as waste, like fecal pellets, and it can also be released by viruses. So again, about half of the organic matter produced by phytoplankton is released to the water, and it becomes dissolved organic matter. But that dissolved organic matter can be consumed by bacterial cells and also protozoans, so single cellular heterotrophs or eukaryotes. So these two processes are the microbial loop. This release of dissolved organic matter and then the consumption of dissolved organic matter. So if we look at a very simplified epipelagic food chain, we have primary producers, primary consumers, and secondary consumers. But what we don't see in this very simple food chain is the microbial loop, this very complicated process that's supporting the base of the food web. So supporting the plankton and the zooplankton. So the dissolved organic matter released from plankton is consumed by these protozoan grazers and can also be consumed by different bacteria like picoplankton and nanoplankton. So rather than lose that dissolved organic matter to the seawater, it's going to be recycled. It's going to be consumed by protozoans and by picoplankton that are going to become a part of this food web on the left, this food chain on the left. So the base of the food web is supported by the microbial loop, which is recycling and using all of that dissolved organic matter. So we've looked at this map a couple of times, this chlorophyll map, to see how nutrient concentrations differ around the globe. But this is an annual average. So if we look at concentrations of chlorophyll and average them over an entire year, we get this image. But productivity can change seasonally. So we see differences in chlorophyll concentrations based on different latitude. But we also see differences in different seasons. The primary production, the production of organic material from inorganic nutrients using sunlight. So this is the result of photosynthesis. Primary production is telling us how much photosynthesis is happening. And it's going to vary based on solar radiation. So seasonally, solar radiation is going to change, and also by the amount of nutrients. So we need sunlight and nutrients for photosynthesis to occur. So variations in sunlight and nutrients across the globe are going to create variations in primary production. Solar radiation varies both with depth and latitude. So we know that sunlight is attenuated and scattered within the upper water column. So we don't see sunlight penetrate deeper than 200 meters in the open ocean, and it's much shallower in coastal waters. And solar radiation is more intense near the tropics. And that's because the tilt of our Earth. So we have the same solar intensity hitting the Earth at the poles as well as the equator. But because of this angle, the same solar intensity at the tropics is focused on a much smaller area than the same solar intensity at the poles. So same amount of sunlight is going in, but near the North Pole and the South Pole, it's spread out over a greater distance. But near the tropics, it's focused on a smaller area. So we see a greater solar intensity near the tropics between 20 degrees north and 20 degrees south, and less intense sunlight near the poles above 60 degrees north and 60 degrees south. And then that's going to change seasonally with Earth's rotation around the sun. Nutrient concentrations vary around the globe. So nutrients, remember, come from the land. Weathering, so wind blowing and water running over the land can deliver nutrients to the ocean. Rivers bring nutrients into the ocean as well as dust. So here we're looking at sea surface phosphate concentrations. We can see upwelling along Peru, upwelling in the Antarctic circumpolar current, bringing higher nutrient concentrations to the surface. Here's a satellite image of Saharan dust blowing across the entire Atlantic Ocean. So we have these variations in nutrient concentrations around the globe, and we can even have these episodic events that bring a lot of nutrients. So this really big dust storm carrying dust across the Atlantic Ocean can cause a phytoplankton bloom. That's an episodic event. So we get variation in solar intensity, variation in nutrient inputs. And remember, we have release of nutrients as we go deeper into the water column. So at the surface, we have higher oxygen concentrations due to photosynthesis. And then as we get deeper into the water column, we see oxygen concentrations decrease as the heterotrophs take over and we have respiration. So respiration is breaking down organic matter, releasing carbon dioxide, but also releasing whatever nutrients were trapped in that organic material. So as we go deeper in the water column, we'll see higher concentrations of nutrients like phosphate and nitrogen. And this is really important because the mixed layer can bring those recycled nutrients back to the surface. So remember, the mixed layer is that upper part of the water column where temperature, salinity, and density is the same. So I'm tracing the mixed layer here around 28 degrees centigrade. And then as we go deeper into the thermocline, temperature is decreasing, and then temperature is pretty stable with depth. The depth of the mixed layer is something that changes seasonally. And we call it a mixed layer because all of this water is going to be mixed together. So no matter how deep the mixed layer is, all of that water is mixed up. It's like taking a bottle and shaking it up and mixing so everything is homogeneous. So in the summer, when we have warmer temperatures, the mixed layer is shallow. So the surface of the ocean heats up. It's a lot warmer than the water underneath, and we have this shallow mixed layer. In the winter, that surface water is going to cool off. So we're at about 28 degrees centigrade here. This water is going to cool off during the winter to about 15 degrees Celsius. And that temperature is going to be closer to the cold water found underneath. So we get this deeper mixing layer in the winter. And this is important because remember, as we get deeper, and I'll flip back to the last slide, as we get deeper in the water column, we have release of nutrients. So when the mixed layer becomes deeper, we start mixing this deeper nutrient water back up to the surface. So those nutrients that are being regenerated down here are going to be mixed up back to the surface, and then primary producers are going to use those nutrients. So now we're going to look at how productivity changes throughout the year in different regions, starting with high latitudes, which are polar regions like the North Pole and the South Pole. And we're looking at changes for different parameters throughout winter, spring, summer, and fall. So the main factor controlling productivity in polar regions is sunlight. Remember, in the winter, we have no sunlight. The sun never rises. So we have 24 hours of darkness. In the spring and the summer, and this example is for the North Pole, in the spring and the summer, we have 24 hours of sunlight. And then in the fall, the sun sets and doesn't come back up. So throughout the year, we have a really big range of light intensity that's hitting polar regions. Nutrients are pretty constant throughout the year, and that's because we have this deep mixed layer. So there's plenty of nutrients, but not enough sunlight during the winter and the fall. In the spring and the summer, when the sunlight returns, we see an increase in phytoplankton and zooplankton abundance. So we see a little dip in nutrients during the summer and the spring because that sunlight and nutrients are being combined for photosynthesis. So this is where we get a little bit of a bloom. In tropical regions, so now we're talking about regions close to the equator. In tropical regions, the main limiting factor is nutrients. So if we look at the surface water temperature throughout the year, there's really not much of a change. So we have warm surface waters, and that creates, remember, a more shallow mixed layer. So the mixed layer in the tropics is not deep enough to bring recycled nutrients back up to the surface. And that remains pretty consistent throughout the year. So nutrient concentrations in the tropics are generally pretty low because we have this very shallow mixed layer. The regenerated nutrients are staying deeper in the water column. They're not brought back up to the surface where they can be used for photosynthesis. There's plenty of sunlight throughout the year, so light is not a limiting factor, but low nutrients are a limiting factor. So we generally see low phytoplankton and zooplankton abundance, and we don't have blooms. So in high latitudes, we get this spring and summer bloom. Tropical latitudes, we don't really see much change throughout the year. Mid-latitudes, so this is the region between the tropics and between the poles, are controlled by a combination of light and nutrients. So we have sunlight year-round in temperate regions, but sunlight intensity increases during summer and spring. Let's just start in the winter. So remember, in the winter, we get a deeper mixed layer. Regenerated or recycled nutrients from deeper below are mixed back up to the surface. So we have lots of nutrients in the winter, but we have lower sunlight intensity, so we don't see a lot of primary production. As we go into the spring leading into summer, we have this increase in light input, and all of these nutrients that have been mixed up to the surface during the winter are going to be consumed, and we're going to get this nice spring bloom. But as we move forward into spring and summer and temperatures increase, we see the depth of the mixed layer move up. So now in the summer, we have a more shallow mixed layer. All of the recycled, regenerated nutrients are down here. They're no longer being mixed up to the surface. So even though we have plenty of sunlight in the summer, we don't have as much nutrients because of this shallow mixed layer. So there's still some primary production taking place, but not as much as the spring. And then we get a second bloom, this fall bloom, as we move from late summer into fall. So even though sunlight intensity is starting to decrease, the mixed layer depth is now getting deeper again. So we're starting to mix up some regenerated nutrients from deep below. So we get this second fall bloom, not as large as the spring bloom. So the spring bloom has all these nutrients that have been mixed up throughout the entire winter. We get a little bump in nutrients around fall due to the mixed layer beginning to deepen again. So the surface water is getting colder, mixed layer is deeper, we have a little bump in nutrients and still enough sunlight to have a fall bloom. That's the end of this lecture. Thank you for listening and watching. I'll post these slides online as well as a short quiz in Canvas, and I'll give you the opportunity to ask any questions that you have about this lecture. Have a great holiday break, and I will see you in class next Monday.